All right. Hello, everyone. My name is Mahmoud Sadeghi. I am one of the co-founders of BIPOC Neuro. I go by he, him pronouns, and I'm excited that you all are here today to hear our speakers from Biogen. This is our second webinar with them in our partnership during the summer, and I will pass it on to Alexa.
Hello, my name is Alexa. I'm going into my junior year of high school, and I'm also one of the co-founders. And I hope you guys are just as excited as I am to be here.
Okay, I'll jump in on the Biden side. I'm Steph Lavin. I go by she, her pronouns. I am an HR partner with Biogen, and myself and Mercedes have been working with Mahmoud and Alexa and the team at BIPOC Neuro over the past couple of months to get this speaker series underway. And we're really excited for the presenters today. Pass over to Mercedes.
Hi everyone, I'm Mercedes Baina. I'm a cellular molecular neuroscientist at Biogen. I've been working with this lovely organization, BIPOC Neuro, alongside Steph, doing these webinars, setting up the webinar series. I go by the pronouns she, her, and I will pass it on to Isha, just to introduce yourself.
Hi, everyone. My name is Isha Mehta. I work on the epidemiology team at Biogen, and I'm very thrilled to be here and excited to talk to all of you. I'll hand it to Sandy.
Hi all, I'm Sandy Engel. I'm a senior director here at Biogen, and I lead our translational cellular sciences group, and I'll talk more about that in a few moments.
Okay, I think we're ready. We can start to jump into the presentation. So Sandy, while you're pulling your slides up, I will kind of your speaker bio. So Dr. Sandy Engel is currently a senior director at Biogen, where she leads the translational cellular sciences group. Her team uses human stem cells, genomic engineering, electrophysiology, and microphysiological systems to develop in vitro models to enable drug discovery for neurological disorders. She received her bachelor's in biology from Ball State University and a PhD in medical and molecular genetics from Indiana University School of Medicine, as well as completing postdoctoral work at the University of Cincinnati School of Medicine. Dr. Engel has over 18 years experience working in the biopharma industry, developing genetically engineered mouse models and in vitro assays. I will turn it over to you, Sandy.
Thank you very much. And can you just confirm that everyone can see my slides? That would be great. Excellent. So I want to thank BIPOC Neuro for inviting me to speak today. I think it's an awesome opportunity to talk to you all and to tell you a little bit about what I do. So the topic, I do need to make this disclaimer. The presenter, me, is a Biogen employee and a shareholder, and the viewpoints that are expressed today are my own. So with that, I was asked to talk about the day in the life of, and as I started thinking about this, I was like, well, what should I talk about the day in the life of? And the first thing that came to mind is the stem cell biologist. And to be fair, I'm going to talk about that. But then I thought, but wait, there's so much more to me because I'm a drug developer, and that is really important to me. Like, I chose to be a drug developer. And then I thought, well, the other thing that I love is a people manager, and you can see some of the folks that I work with on the side, and I really, truly enjoy the opportunity to work with a wide variety of people. And I thought I could spend, 20 minutes talking about how important that role is. And then I thought, well, but I'm also a daughter and a sister and an aunt and a wife and a pet parent and a square dancer, which I could also spend 20 minutes on any one of those topics and how important they are to me. But I thought probably what you were all looking for is that sort of first thing, which is, let's talk about a stem cell biologist, because I will say this is something I'm super passionate about. It's probably the coolest thing I could do. It's just short of being a drug discoverer. And I feel like this is something that might be of interest to you all. And then I thought, well, if I'm going to tell you about the day in the life of, I really kind of have to start with a longer journey, because it's not like I just showed up today and I get to do this really cool job with these really cool people. I actually took a lot to get here, and it's that lot to get here that really helped me be who I am and sort of helped me find this perfect job for me. And so you know, it's a journey, and we all take a journey. And mine started in Idaho Falls, Idaho, which was where I was born. But my parents are from upstate New York. And so very shortly after that, we moved back to upstate New York to a town called Queens Hollow. And I point this out because Queens Hollow sounds just like it is, a teeny tiny town in the middle of nowhere in upstate New York. And we actually lived on the edge of my grandfather's dairy farm. And I point this out because nothing in that start would make you think that I would be doing this today, because, you know, my parents, my mom's parents were farmers and my dad's parents were factory workers, and there was nothing to suggest that I would become a scientist. And then we took a deep dive to Cali, Colombia, because my dad was a chemist and he wanted to do something fun and interesting. So he went and took an overseas opportunity in Colombia, and then we moved on to San Jose, Costa Rica, and then back to Queens Hollow, and then over to Ankeny, Iowa. And then I point out Indiana because Indiana kind of got there for a while, and so moved to this tiny town in Indiana called Rosedale, Indiana. It's just north of Terre Haute, Indiana, which Terre Haute has little claim to fame, except that that is where Larry Bird got his start as a basketball player. And I point out Rosedale because, again, those rural roots of my parents, this town was 500 people total. In my high school, there were 200 people. Not 200 people in my graduating class, but 200 people in my high school. There were 50 folks in my graduating class, and there was one English teacher, one math teacher, one science teacher. So again, nothing in this would have argued that I was on a path to doing all of these awesome things that I get to do. So I'll mention Muncie, Indiana, and Indianapolis. They have stars. I'll come back to those in the next slide. Similarly, from there, I moved on to Burlington, Kentucky. And then we just jumped out of this for no good reason. Let me see if I can get back. So Burlington, Kentucky. From there, Phillipsburg, New Jersey, Ledyard, Connecticut, a lovely place. Poway, California, and now I'm back here in Norfolk, Massachusetts. And so what I just wanted to point out is that it is all these different experiences where I've gotten the opportunity to work with really great people, to get to know really great people, to get to experience different parts of the country. And this to me has been really important in helping me understand who I am and what I want to do and what makes me happy in helping people. So I mentioned I wanted to highlight Muncie, Indiana, Indianapolis, and the University of Cincinnati. And I pointed out already Rosedale. So I think that, you know, for most of you right now, your education is sort of the thing that's taking up most of your time and attention. And these are really transformative moments where you learn a lot about what you think about the world. And so I did want to just take a brief moment to talk a little bit about some of those big lessons I learned in different places. So the one I learned in Rosedale is that you can come from a very small place and you can, you know, go on to do bigger and better things. One of those things is that when I went off to college, like many people, my family didn't have a ton of money, and I was the second daughter. And so there wasn't much money set aside for me to go to school. And I will say this is the first thing I learned, which is you should ask for help, because it was the help of people within my school system who helped me identify a college. It was talking to the college about the fact that I didn't have a lot of money to go to school, who helped me find scholarships in order to be able to go to school. And that was really important to me. And so I encourage everybody to seek help, and to seek help often, because it is really relying on other people that make a difference. And then I will say the other thing I learned is to say yes. And it has an asterisk behind it, because you should say yes when you can, not to everything that happens. But I will say that one of the things that happened when I was an undergraduate student is somebody asked, you know, we have this job as a research assistant in the Muncie Med Ed Center. Would you be interested in it? At that time, I had about 3 lab classes under my belt, and I thought, there's no way I am qualified to be a research assistant. But somebody said, what's the worst that can happen? They can fire you? And I'm like, true. So I said yes, I got the job, and that was one of the most pivotal things in my life, because I found out how really wonderful it was to be in a research environment, to work closely on people, or closely with people on hard problems related to science. And if I hadn't done that, I might have missed out on this entire career in research. And then you give what you get. Probably nobody on this line but me knows about Ball State University. If you've ever drank out of a ball jar, like a canning jar, that's them, the Ball brothers. They developed a teaching college because they felt like Indiana needed more teachers. So clearly Ball State University wasn't meant to be like a high-powered research university and not in science at all. But if you go somewhere and you put your effort into it, you can get a good education no matter where you go. And I will say this is where I learned that human biology is endlessly fascinating. I was fascinated by biology when I was dissecting, earthworms in my biology class in high school. And when I got to a place where I learned about human biology and human genetics, I'm like, this is what I want to do for the rest of my life. And I was really lucky because, again, I asked for help. And then when I got towards the end of my college career, they were like, well, they're at Indiana University, they have a whole program in human genetics. Like, you could just do nothing but study this. And I was in heaven. And so I went to Indiana University as a PhD student, and I didn't know anything at all about graduate school. Because remember, I mentioned that my parents had been the first ones in their families to go to college. So for me, graduate school was like, I'd never even heard of it, never met anyone who went to graduate school. So again, getting help from people who knew and who could help guide me was really important. So I entered as a graduate student, and the first thing I learned was that working with humans as an experimental model is really, really hard. Like, a generation takes 25 years, and people don't breed the way you want them to do good genetic experiments. It's just very frustrating. And this is where I learned that mouse models could be a great substitute for human biology. And this is also where I fell in love with pluripotent stem cells, and I'll talk about them next slide. I mentioned the University of Cincinnati because at the University of Cincinnati, this is where I really started to figure out what mattered to me. And one of the things that really matters to me is variety in science. I am so excited about lots of different kinds of science. And so when I would meet my fellow colleagues who were like, I'm a cardiac scientist, or I'm a brain scientist, I was always like, but then why would you not get to, if you were a neuroscience, then you miss out on all the cool things the heart does. And if you're a cardiac scientist, then you miss out on all the cool things that the skeletal muscle scientists work on. And so for me, I really like a place where I can interact with as much variety of science as possible. The other thing I learned is that I really, really like helping other people. For an unfortunate situation, I accidentally got to lead their mouse making group, which was really cool. And I got to work with scientists all over the company or all over the college. And I really enjoyed that interaction and that being able to help them do what they wanted to do. And I liked the accountability with it. Like, people came to you and they asked you to do something and you gave them a timeline, and then they wanted it when you said that. And I really liked the challenge of being able to do that. And then I also really liked my role of having a purpose. I really felt like what I was doing is important, and that's been a huge driver for the work that I've been doing going forward. So that is my background. So now I want to talk a little bit about science. So I mentioned that Indiana University is where I fell in love with pluripotent stem cells. And for those of you who haven't been hanging out in the stem cell world, you're probably asking, well, what is the stem cell exactly? So let me tell you a little about these really cool cells. So stem cells are all over your body, and there are different kinds of them. And so there are ones that are called totipotent stem cells, and that's the fertilized egg. And it's totipotent because it can make every single cell of your body and every single cell that's needed to make your body. And that's the things like the placenta and things like that to actually have a body form. And so there are a handful of cells that are totipotent, can make everything. But then once the embryo starts to form, then things start to narrow down. They can't make everything anymore, but they can make a lot of things. And I want to point out these cells that are called from the inner cell mass of a blastocyst. So just 4 or 5 days after an embryo is fertilized and it starts to form, there are these really cool cells that form. And these cells are pluripotent, which means they can form everything that makes the embryo. The only thing they can't form are the things that go on to make the placenta. And these cells are awesome, so keep that in mind. There's also some other cells like germ cells, which also can make pretty much every cell in the body. But then, interestingly enough, once you're born, everything's called an adult stem cell. And this just means that it's a fully formed tissue. And there are ones that are multipotent. And you guys probably know a little bit about multipotent stem cells because these are the kinds of things that are sort of familiar to you. You probably heard about cord blood cells that you can get from an umbilical cord. these are considered multipotent because they can form a lot of different things like blood cells, and you can kind of tweak them to form some other things, but it's kind of limited what they can do. And you know about cells in your gut, you know that your intestine is always proliferating, and those are, again, adult stem cells that do that. So that's really cool. And they're tissue specific because they only make one sort of handful of cell types. Interestingly enough, if you think about cancer, cancer is just sort of uncontrolled cell growth, but cancer stem cells are also a kind of stem cell because they can make themselves, because there are two key properties of stem cells. Stem cells can repopulate themselves and they can differentiate into something else. So they have to have those two properties to be a stem cell. And cancer stem cells can do those things. They can make more of themselves and they can differentiate a little bit. So they get to be stem cells too. And then I will talk about induced pluripotent cells in a moment, because those cells just changed biology. I do want to point out and come back to these embryonic stem cells, because this is where I fell in love. When I found out that you could take mouse embryonic stem cells and grow them in a dish, and these cells, these pretty little colonies of bunches of cells there, and then you could put them back into a mouse blastocyst and you could form a whole mouse embryo, that was awesome, because here were things in a dish that you could turn into a living organism in your hand. And the beauty of these cells is that you can change them. You can add genes, you can take away genes. And so now for the first time, you had this ability to take and manipulate something and then make it a living organism to ask questions that you couldn't ask previously. But today I'm going to talk about human-induced pluripotent stem cells, because these are the same thing from a mouse, but now they're from a human. And the cool part about these cells is that they start from an adult. So this means that you can start knowing something about the person that you're getting the material from. So you can pick the person. And what you can do is you can, let me go backwards. What you can do is that you can take cells from that person, and it started out being fibroblasts, so things that come from your skin, but now we can do it with cells that come from your blood, or we can do it from cells that we extract out of the urine, or we can do it from your hair cells. But we can take those cells, and by adding in some genes, we can revert them back to that pluripotent state, that state that resembles what happens from the inner cell mass, so they can make all the cells of your body. And the key point here is that because we start with an adult, we know about that adult. We know whether they got a disease or they didn't get a disease. We know whether they responded to a drug or they didn't respond to a drug, or we can select people who are ethnically or genetically diverse if we're trying to understand sort of how populations respond to things. And this is powerful, because before we had this, we really had no way to model humans. But now we have what I started out in my career doing. I wanted to model human genetic disease, and now I could actually model human genetic disease without having to worry about all of those things like 25-year lifespans. And the cool part about stem cells that I talked about is that they have to be able to make themselves, which is really important, because if they can make themselves, then you can make huge quantities of them. If they can make themselves, then you have the opportunity to go in and genetically modify them, just like I did with the mice. And then the other property is they have to be able to differentiate into things you care about. And when you look here, I can take those cells that are undifferentiated and I can turn them into things that I care about, like skeletal muscle cells and blood cells and adipocytes. And here at Biogen, what we like to work on is neurons. And so this is really cool, because now I'm able to model human genetic disease in a dish. And when I can do that,
I would just like to say we have 5 minutes left.
Okay, I will finish up. So when I can do that, that allows me to think about developing new drugs. It allows me to think about developing new theories. So Translational Cellular Sciences, the group that I lead, we do 4 things. We develop stem cells, which I just talked about. We can modify those stem cells. We can characterize those stem cells with electrophysiology, and then we can build assays with them to help us do drug discovery. And so I'm going to just do a quick walk into the laboratory. We're all suited up to go on in, And I'm going to talk about one example of how we apply this. So many of you may recognize amyotrophic lateral sclerosis. This is known as Lou Gehrig's disease, and many of you probably recognize it from the Ice Bucket Challenge. This is a devastating neurological disorder. It's progressive, and it usually leads to the loss of motor control. And usually after you're diagnosed, you only have about 3 to 5 years before the disease takes over. Now, there are two types. There's sporadic and there's genetic, and most of it is sporadic. We don't know what causes it, but for about 10%, we do know that it is caused by specific genes, and about 8% is caused by this gene called C9orf72. And the thing I just want to point out here is that one of the things that we know happens is that it forms these RNA foci that we can see in cells. And so my job and the job of my group is to really try to model that in a dish. So the way we do that is we start with our induced pluripotent stem cells from patients, and then we differentiate them through a series of steps to make neurons in a dish. And then we can stain those neurons in a dish, so they're behind me and in this picture. And then we can use technologies to ask how close are we in making the cells that we find in the body. And then we can ask whether they actually mimic the disease. So here on this left side are ones from a patient that, or from an individual who's wild type, who doesn't have the expanded repeat. And what you can see over here on the right side is those RNA foci that I mentioned. We can see those form in a dish in a patient who has that expanded repeat. And because it's a disease, it's called a neuromuscular disorder. It's neurons interacting with skeletal muscle. So we have to be able to make skeletal muscle as well in a dish. And then we can actually mimic that entire process of making neurons, having the neurons grow, and making a little tiny skeletal muscle bundle in this little tiny microwell. And you can see that the neurons interact with the skeletal muscle. And then hopefully you can see over here on the far side is the contracting muscle bundle that's coming from the neurons that are innervating it. And so that is a really quick walk through my day and the kind of cool science that I do. So I do want to just remind people that, so what does my life look like? What does a day in my life look like? It looks like a lot of cool science where we get to work on the cutting edge of what's going on in the scientific world. I always tell my group we are problem solvers. We spend our time observing, evaluating, making hypotheses, testing and iterating around that to address problems in drug discovery. It's about being a team player. Everything I do is within this awesome team of people within my group and within the larger Biogen community. And it is getting to interact with these really smart people doing really important things that are really gratifying for me. It's about optimism. We have to be optimistic. 9 times out of 10, the work that we do, the experiment we did doesn't work. And so we have to be optimistic and confident that to next time we try it, we'll have thought about the problem and we'll have figured out how to solve it. And if we do the experiment and it still doesn't work the way we think it is, we come back and try it again. And we believe that we're making a difference for these people. So with that, I'd like to just really thank all of the folks, because as I said, this is a team effort, and it is the ability to work with that team and help each other that really drives my day. And so with that, I'm happy to stop and take any questions that people have.
Thank you so much for your presentation. Attendees can use the chat function to ask questions or ask to unmute.
While people are thinking, I'll ask a question. Sandy, how do you get patient cells?
That's a really great question. So we work with a lot of the hospitals in the area. And so they are the ones who see the patients and know of the patients. And so we partner with them. We partner with academic groups who see patients. And then these are such valuable cells that lots of people are generating them. So now there are actually IPS banks where you can just go and purchase them. And so it makes it easy to work with these cells. So I see that there's a question in the chat box about how the cells are used in drug development. And so for us, we use these cells to help us develop in vitro assays. So, oftentimes we need to test a lot of things before we can find the one that's actually going to act as the drug. And so if we can do that in vitro in a cell culture system, then we don't have to use animals and we don't have to worry about patients themselves, and it helps us narrow them down. So we really use these a lot to build assays where we can test compounds, and so help try to pick the best compound to move forward as the drug. And so how can students get involved in research? I see that question. I think one of the things that you can do is as you go through school and get involved in college, reach out to your professors and ask them if they're involved in research and if they'd like someone to help them. You'd be surprised, in many organizations, there are undergraduate positions where you can work in a laboratory and become familiar with how laboratories work. So I really encourage you, if you are going to college and you have a professor that you really appreciate what they're doing and how they teach, approach them and ask them if they have a research position or if they know of someone who does, because that's how I got my start. And if I hadn't had that opportunity, I would not have known how awesome it is to work in a laboratory. And then there's a question about looking back on past failures. Is there a specific method of thinking that ultimately leads to final success? I don't know that there's one specific way to do that, to get to ultimate success, but I do encourage people to accept that failure is part of the process. In a short presentation, you get to hit the highlights and the things that worked. You don't get to dwell upon the fact that there are plenty of times where you spent 6 months trying to get something to work, and in the end, just never did. And that's why I encourage people to, be optimistic and have faith in yourself, because you have to sort of believe that, okay, the next time, like I've thought about it, like I figured it out. And so the next time I do this experiment, this is the time that it's going to work. And I will say the time that it does work, you can celebrate and everybody in the group with you will celebrate. And that's a really great feeling. And then, does your lab use HTS in the drug discovery process? And if so, has your lab produced drugs in a recent or past years? And so I will say that I think this is the holy grail of everyone who does drug discovery, is to be involved in a project that makes its way all the way from the beginning of where they're involved to the clinic where it's actually helping patients. And so I will say I have worked on lots of projects and done little pieces of them, some of which made it further than others, but I can't say that I was instrumental in driving any single project to the market. But I don't think that, for me, that's the critical driver. For me, I know that there are patients out there that need help. And I know that even if my cell assay is the very first thing that happens in this process, if my cell assay doesn't happen, the process never goes anywhere. So even though that by the time something makes it to market, me and my folks haven't touched it for 8 years, it doesn't matter, because we did our part. We helped it get that far. And even when we work on projects that die, I never feel bad, because our assay helped determine whether we should move that forward or not. And if ours helps say that it shouldn't go forward, then I'm as proud as if I helped something get all the way to the market, because it is the process of science that we are helping to move forward. So how long is a typical day in your life when you get up and when you go to sleep? So I am a morning person. I just want to say that out front. So I typically get up somewhere between 3:30 and 4 a.m. And I typically go to bed around 10 o'clock, but the 10 o'clock is pretty a hard stop. My husband jokes because I'm pretty much asleep by like 9:45. Don't ask me tough questions. So, and I think that's just who you are, and sort of I joke that I have a very strong circadian rhythm. So like I get up, I'm good, I fall asleep. So. A good question about how does industry lab differ from academic lab? So I will say that having been an academic lab, for me, part of the challenge in an academic lab is that the pace is kind of slow. And there didn't seem to be a whole lot of accountability. I knew people who kind of meandered through the work that they did, and everybody seemed fine with it. And that's why I said, for me, I learned that I like accountability. I like it when when someone sets a goal, we set a goal and we try to achieve it. That's very motivating for me. So, how does it differ? I think we have a clear understanding of what we're trying to do. And there's an expectation that we will do it. And one of the things I really love about industry science is that we do know there are people at the other end. So we have to do our science to the highest level. When somebody builds an assay, that assay will run the same way for everybody, because if we're going to use it to make decisions about people, we want to make sure it's right. And so for me, those are the kinds of things that make it super exciting to work in industry. I can clearly see the patient at the end. I know that we're going to do the best science we can because we know there are patients at the end. And I like the fact that we are working in a team towards a common goal. Those are really important for me.
Thank
In
you,
your
Sandy.
opinion, oops, sorry.
I know people might have more questions, but I wanted to give time for each to speak, and then whatever
Absolutely.
remaining questions we have, we can answer after.
Okay, thank you.
Isha, do you want to, I can read your bio while while you're pulling your slides?
Sure.
All right. Isha Mehta is currently an epidemiology research manager at Biogen. She received her bachelor's from Purdue University in brain and behavioral science and a master of public health from Dartmouth College. Her professional career has focused on a variety of neurological and neuropsychological conditions ranging from alcohol and opioid addiction to chronic neurodegenerative diseases. She's particularly passionate about increasing diversity by race, ethnicity, age, and sex in both STEM research personnel and clinical studies.
Awesome. Am I on mute?
We can hear you.
Oh, you can hear me. Sorry, I couldn't figure out where the little bar went. Hi, everyone. My name is Isha Mehta. I'm so excited to be here today and talk with you all. I work on the epidemiology team here at Biogen. I will warn you, I talk a little quickly, so feel free to drop in the chat if you need me to repeat something. And I use my hands a lot. I'm a very animated person, so just bear with me through that. So yeah, we can get started. So little disclaimer at the beginning. So, I am currently an employee and shareholder of Biogen. Any of the viewpoints I express today are strictly my own and in no way reflect the official position or views of Biogen. So I want to challenge a couple of assumptions that you might have about industry scientists. What do you picture when what does a classic industry scientist look like to you? Are they wearing a lab coat? Are they using a microscope or some other sort of technical piece of hardware? Are they wearing lab goggles? What even physically do they look like? Are they a certain demographic? What race are they? What ethnicity are they? Are they male? Are they female? What do they look like? And I think for a long time there was this, bias or this belief that, scientists or, even industry scientists were sort of middle-aged white men. And as I think you can see from Sandy and I, that is not the case anymore. And that's really awesome. We're really seeing a lot of diversity in industry science, and it's about time, and it's great to see that. So I am not somebody who wears a lab coat every day. I do not go into a lab. I do not use technical hardware in that way, but I am still considered an industry scientist. So industry scientists work on all ranges of the spectrum. So some study organisms that are invisible to the naked eye, and that work is really important. But others like me study entire populations, entire countries. So, the work that we do and the sample sizes that we use are thousands and millions of patients, which leads me to epidemiology, which is the specific field that I work in. So what is an epidemiologist? So since COVID started, epidemiology has become a much more mainstream word, but I still don't think there's a great grasp on what exactly, epidemiology is. So at a very sort of basic level, epidemiologists are public health experts and advocates. We are scientists who study diseases within specified populations within specified sets of time. So some epidemiologists focus on infectious diseases like COVID-19, and that work is very important and very necessary. But others like myself, we focus on complex neurological diseases, whether they be chronic or acute. So what do I do every day? That all sounds great, but at a given time, I am focused on a specific disease area. So my daily work revolves around furthering our understanding about how a disease manifests, progresses, and even maybe disappears in certain populations. If we're talking about, neurodegenerative, chronic diseases, those are not going to just suddenly disappear. But if we're talking about acute events such as, maybe a heart attack or something like that, then that is, that's not a persistent state. So that is a whole, subset of epidemiology, is understanding, when those diseases disappear, are they going to reappear? What's the likelihood of that? So let's take a look at an example. So somebody, in the team might come to me with, a hypothetical question. Which of these two people is more likely to develop Parkinson's disease, a 70-year-old white man or a 45-year-old Asian woman? So at a surface level, there's a lot of ways to approach this, and it's not exactly a clear black and white, answer. And so first, we know that Parkinson's disease is an age-specific disease that primarily affects the elderly. Now, we know this because of past epidemiologic work that has been done, and we are able to say with a degree of certainty that is true, that it mainly affects elderly patients. Second, these long-term observational studies tell us that Parkinson's disease mainly affects white people. So the type of data I work with is observational, meaning they're non-interventional. So I am just observing how a disease or a condition or an issue, affects a population over time. I'm not intervening in any way. I'm not doing anything that would not have already happened naturally. So those studies tell us that Parkinson's disease mainly affects white people. And by long term, I mean they're generally years, sometimes decades long. And third, these long-term observational studies tell us that Parkinson's disease mainly affects men. So through sort of process of elimination, or rather sort of confirmation of all of these different variables, I'm able to come to the conclusion that between these two people that I presented earlier, a 70-year-old white man and a 45-year-old Asian woman, a 70-year-old white man is more likely to develop Parkinson's disease at that specific moment in time, not considering if the Asian woman ages. So that's great. Now what do I do with this information? I know that white men are at a greater risk for developing Parkinson's disease. That's wonderful to know, but what do I do with it? Well, a lot of things. So like any scientist will tell you, you have to confirm. Just because you have that finding does not mean that you can run with it. We would confirm against if there's already published information on that, or if it's new information and we have, a degree of confidence with it, then we would go ahead and publish. And this has a lot of interesting and really important implications. So not only can it introduce new avenues of research, but as I mentioned before, epidemiology and epidemiologists, we really are public health experts, and advocates. So the data that we publish has public health implications. And so this data can really go right into a clinic. It can really affect conversations between a patient and their provider, a provider and caregivers, provider and families. Perhaps a provider will realize that one of their patients is at a substantially higher risk of developing X disease than another one, and that could really inform their treatment and their recommendation to these patients. So it starts at a very sort of population level, but it can come down to individual care levels too. And so if we're using the example of Parkinson's disease, then this also has implications on Parkinson's disease clinical trials. So, we would want to make sure that we have proper representation in these clinical trials. We want to make sure that we, while it's important to be diverse, we want to make sure that the right people are included in our clinical trials. So there are a couple of historical examples of this where, there's been a problem when we haven't had the right people in studies. So for example, heart attacks in women. So for a long time, it was thought that women just didn't have heart attacks, right? Because they didn't present with the classic chest pain. It was mostly men who were presenting with that, and they just thought, women couldn't have heart attacks. There was some sort of innate reason for that. And it was through studies like the Framingham Heart Study and other long-term observational studies that we realized women are having heart attacks, women are dying from heart attacks, but their manifestation of those heart attacks is different than the manifestation in men. You know, women have gastrointestinal issues when they're, you know, that's the classic sign of a heart attack in a woman. And so we were able to develop interventions. You know, doctors learned from that of, you know, if a woman of a certain age category comes in presenting with gastrointestinal issues, you know, I need to look out for, is she having a heart attack? But we wouldn't have known that had it not been for those long-term, you know, natural history of the disease studies. Another classic example is, you know, sickle cell anemia. It was through these studies that we realized that, you know, a majority of the sickle cell anemia population is African American. You know, we took a really long time to understand that, but through this, you know, these observational studies and naturally observing how a disease progresses, that we were able to come to that conclusion. So all of this can help ensure that the right medicines go to the right patients. So tools of the trade. So I sort of use what are considered maybe a little bit less traditional sort of tools in the way that I pursue science. So like I said, I rely on data from observational population-based studies. So population-based means that, you know, it's from a sample of people that the results could be generalizable or applied to the rest of that population, whether it be the rest of that country, the rest of that demography, you know, whatever it might be. So ideally, these are huge studies. You know, they can have hundreds of patients, maybe even thousands of patients. So in my line of work, the more patients, the better. You know, we want as many people as we can, because that means it might be more generalizable. You know, it'll help us understand the condition more. I also use what are considered these big data resources. So big data has become much more of sort of like a hot term, you know, in recent years, but the resources have been around for a while. So these data sources include CDC studies. So the CDC will, you know, have long-term observational studies that they routinely collect across the nation, you know, for years and years on end. Some go back all the way to the 70s, and you know, they're continuing on now. And these are publicly available data sets. You know, anybody can go in and, you know, do their analysis with this. You know, of course, you do need to have a little bit of sort of some programming skills, and you do need to read a lot of sort of what are called data dictionaries, but it's available for anybody to use. We use Medicare and Medicaid files. So Medicare and Medicaid, as some of you might know, are governmental insurance programs. But in addition, those programs, so CMS, also collects data on the enrollees in Medicare and Medicaid. So I should let you know that it's completely de-identified, it's completely anonymized. There is absolutely no way that somebody like me or another sort of epidemiologist or scientist could go in and, you know, pinpoint an exact patient record to, you know, a specific person. It's not like that at all. You know, it's aggregated. So, you know, CMS might tell us, you know, these events happened for women aged 20 to 45. And I might be able to say, okay, I'm looking for a specific trend for a specific disease. You know, are women ages 20 to 45 more likely to break their arm than men ages 20 to 45? And that, you know, I can use that data set to say this is representative of the US population and make conclusions from that. Furthermore, I also use electronic health record data. So electronic health record data is really cool in the sense that, so if you think about whenever you go to the doctor, right? Let's say I broke my arm, right? I go to the doctor and he puts in his file, you know, on May 1st, 2021, Isha broke her arm, right? And then let's say a couple of years in the future, you know, I break my other arm. I'm just not having good luck. I break my other arm. And he'll put in there, you know, 3 years later, you know, Isha broke her other arm. Now, somebody like me might get access to this electronic health record data. And again, it's completely de-identified. There was no way that, you know, a scientist would be able to look at that and be like, that's Isha's record. And could look in, you know, sort of an aggregate way of, you know, maybe women, you know, maybe my question is, are women in their 20s more likely to break both of their arms than men in their 20s? You know, and I can use that to look at, you know, thousands of, or sometimes even millions of patient records and say, you know, with a, you know, degree of certainty, you know, women with women in their 20s are, significantly more likely to break both of their arms than men in their 20s. And this could have all sorts of implications. It could, new avenues of research. Maybe women in their 20s have poor bone density. Are they exposed to more dangerous situations? Do doctors need to warn their patients that, hey, we're noticing you're at a really increased risk of breaking both your arms? You need to be aware. So there's a lot of really useful information that can be drawn from these big data resources. And each of these particular data sources can contain millions of patient files. So it can be really generalizable to the rest of the population. And so tying this back to neurological conditions, this allows me to track and understand neurological conditions at a population level over time. So a little bit about my path and how I got here. So at least for me, and it wasn't that long ago, getting involved in neuroscience, I was really interested in it, but it was sort of a little daunting. Like, what was the way I wanted to, approach it? I knew I wanted to enter the field and make a difference, even if it was in a really minute way. I knew I wanted to have some impact on it. So I got my bachelor's degree in brain and behavioral science from Purdue University, which is a rival to Sandy's school, but we won't tell her. And then I went and I got my Master of Public Health from Dartmouth College. So professionally, I think the best piece of advice that somebody gave me, and I would love to pass it on to you, is you don't need to have it all figured out right away. Some people go to school and they know exactly, what their passion is, what they want to pursue. I didn't know that. I just sort of knew in general, what I was interested in. So start with internships, start with research projects, be a research assistant, shadow people. The other piece of advice I would have is don't feel like you need to jump right into a doctoral program. A doctoral program it's a big time commitment. It's a big energy, commitment. So take the time to figure out what aspects of neuroscience are most interesting to you. And I think being an industry scientist, that's something that's really special, is that you are able to be in an environment where not only are you working on something that you're knowledgeable about and feel like you can make a difference, but you get the chance to meet all these people who are sort of experts in their own right and learn from them and figure out, oh, that's something that I'm really interested in, I want to pursue, or what, maybe that's not really my thing. And so it was through, doing research projects in college that I realized, I'm not somebody who really enjoys lab work. I really am interested in neuroscience, but that is not the avenue for me. But you're not able to figure that out unless you sort of test out all these different things.
Hello, I just want to say we have 5 minutes left.
Okay, thank you. Almost done. And so just to wrap up and summarize, so not every industry scientist works in a lab. So if that's sort of like something you're a little bit iffy on, that's totally fine. There's a lot of us that are really interested in it, and that's just not what we're cut out for. And there's so many elements to neuroscience. So take your time, test them out. Learn from people who have taken different paths than you and have studied different things than you. And I've also thrown up my my contact information up there. It wasn't actually that long ago that I was in school, and I actually started at Biogen when I was still in grad school. And so I've been here for a few years now. And so, I'm happy to, field any questions or just listen to ideas or anything you want. So throwing that up there, but yeah, I'm happy to take any questions that you guys might have. Thank you.
Thank you for your presentation. I believe we had one question as people type other questions from Asha. In your opinion, what was the biggest change you had to adapt to when you first came into the industry?
Let's see, biggest change when you first, so I think you're knowledgeable, but I think you also need to understand that there are people that are way more knowledgeable than you, and it is totally okay to just sit there and absorb all of their knowledge. And I think that was the biggest change for me, is you sort of come out of this academic setting feeling, oh man, I did so great, I finished this project, I did that. It's okay to go in and just be a sponge and learn it all, and you will make mistakes, you will screw it up, that's okay. And so just be open about that, communicate, learn. And so yeah, I didn't know if Sandy had a different answer to that.
I actually think your answer is awesome, because I think you mentioned something that, again, I think is really important, which is the joy of industry is you get to meet so many people who are so good at what they do. And I always joke that I don't need to be a medicinal chemist. I just need to have one as a friend. I don't need to be an epidemiologist. I just need to know Isha, right? So for me, that's the cool part, is that everybody brings their expertise to the table. And that's what I really, really like.
Yeah, you get to learn from some of the best out there. It's awesome. So yeah.
Looks like there's a couple more questions in the chat for you, Isha. So how did you first gain interest in epidemiology?
So it was actually kind of interesting. So I went into, I'm a little wordy, so just bear with me for a second. I went and got my master's degree right at the height of the opioid epidemic. And I had gone to school in the Midwest, and we were really, the opioid epidemic had really hit the Midwest really hard. And so I went and, was getting my degree in public health, and was given, so you had the opportunity to pick a concentration at that time. And, epidemiology was just something that was like really interesting to me, understanding we were looking at the opioid epidemic, and like, for some of us who were, we were young, and we were like, how could this happen? This is brand new. This has never happened before. And we learned that it's not a brand new thing. We have had drug epidemics before, and it was sort of through that lens that I got introduced to epidemiology and understanding, we can really learn how a disease behaves over time. Even, not just a disease, there's, a subset of epi called pharmacoepi. That's understanding how a drug affects a population over time. And so that's sort of how I, got my interest in epidemiology. As to how I joined the Biogen epidemiology team, I actually started as an intern here, and liked it so much, and they couldn't get rid of me. And so I've been here ever since. So yeah.
How does the current epidemic influence your job?
Yeah, so I think that's an awesome question. I think there was a lot of, frankly, it was really frustrating at first, right? There was a lot of misinformation that was out there. We sort of knew that there were things that needed to happen, and this was epidemiology, collecting data that was happening in real time, and that's not a sort of a situation that you want. We had to learn on the fly. And so I don't work in infectious disease, and so infectious disease epidemiology, mad respect to them, because they've had to go through a lot recently. But understanding the concepts, and also the other thing is like being able to be somebody who can just sort of be a source for information for the people around you. And so, we even had conversations at Biogen. I was talking to, I think it was someone in HR, and, pretty early on during the pandemic, just, sort of having a frank conversation about, what are the things they need to look out for. It can also sort of inform conversations about vaccinations. So you're able to sort of be that person in your personal life of like, I can be a source of information for you. Typical.
Yeah, go ahead. I was just going to read the question.
Typical day in my life. So the nice thing about sort of like not having to like, my data sets are on my computer, right? Or they're on someone else's computer, and they email them to me. So I've been lucky that I have a lot of flexibility in my day job. And so, some days, I'm up at the crack of dawn because I'm like, I really just want to answer this question, and I just really want to take time to explore this data, before other people log on. Other times, sometimes I'm not like advocating for this, but sometimes it happens. Like you roll into work at like 9:45, and like, maybe you work a longer day that day. The other sort of plus side is that I get to work a lot with international teams. So, sometimes I'll have 9 p.m. phone calls, and, that you adjust your day accordingly. But I've been lucky that I have a lot of flexibility in the sense that if you have the data sort of at your fingertips, you don't have to go to the data, then it's a lot easier in that sense. Yeah. I'm not advocating for like, don't go to work on time. I'm not like advocating for not doing that, but like work it out with your boss. So, what tools do you use to analyze data, and how do you draw a conclusion from the data? So those are great questions. So the tools, you know, a lot of the time, sometimes it can be something as simple as just Excel. You know, when, you know, inputting data, you know, creating those graphs and those figures. Other times with these larger data sets, it's a bit more complex. So you would use statistical programs. So things such as R and SAS or Stata. And so that's a whole subset. You know, we have a whole team of analysts that work on that. We're all trained in how to use them, but you know, we have great people who do this for a living and, you know, are able to understand the ins and outs and, you know, analyze that data for us. And so the second part of the question is, how do you draw a conclusion from the data? So there's also a heavy emphasis on statistics in this. So I think during the presentation, I mentioned something called significance, or, you know, you might have a significant finding. So that actually has a very particular meaning. And so, you know, it's not that you're just seeing, you know, an X% increase, you know, that significance level lets us know that like the effect or the thing that we're seeing is not by chance, right? There's a reasonable, you can say with reasonable confidence that the result you're seeing is because of X, Y, you know, and then it's not like a random sort of thing that's happening there. How do you know, other ways you draw conclusions? There's a whole other set of statistics involved there. They each have pros and cons, and it's kind of cool. You get to talk about them with statisticians, with the analysts. You get to see, you know, what is the statistic that's going to best fit your data set. And it's very much following the traditional scientific method, right, of hypothesis generation methods, all that sort of stuff. But you know, perhaps it's not as cut and dry. You know, each time it is tailored a little bit.
All right, thank you. I believe we have one more question, but before that, I wanted to drop the survey link in the chat, seeing as it is 4 o'clock Central Time, 5 in Eastern Time. So there is the survey link for our webinar. And Isha, if you have extra time to answer a few more questions people
Sure.
would like.
Yeah.
I want to thank everyone for coming. If you have more questions, please feel free to stay. I just wanted to put the survey link out now, which I will also email to the attendees in one day, and give you all time to answer that to win some Biogen merch. Yeah, I believe that is it.